Welcome to day 2 of our clinical trials webinar. My name is Maria Smith, and I work with the ALS Association in our Mid-Atlantic territory. Thank you for joining us for day 2 of ALS Clinical Research Week. There are a few things we want you to know before we begin. There will be time at the end of the webinar for questions, and you should see on your screens an option for Q and A. You can type questions you have there at any time, and we will review them at the end of the webinar to provide answers. You can also submit your questions in our chat box feature. Again, we will review questions at the end of today's presentation. We do have a very short survey we are asking that everyone complete after the webinar. The survey will appear in your browser once the webinar ends. These surveys help us to know your thoughts on this webinar so that we can improve in the future. For those who are joining us for the first time this week, ALS Clinical Research Week features a webinar every day at 3 p.m. Eastern Time. Each webinar focuses on a different aspect of ALS research. For today's webinar, we are joined by Dr. Nicholas Maragakis. Dr. Maragakis is the Medical Director of the Johns Hopkins ALS Clinical Trials Unit, Director of the ALS Center for Cell Therapy and Regeneration Research, and Professor of Neurology at Johns Hopkins University. The Center for ALS Specialty Care at Johns Hopkins is a world-recognized leader in providing medical care and offering the latest in clinical trials and therapies to ALS patients. Dr. Maragakis received his Bachelor's of Science degree and medical degree from the University of Utah and completed his residency and fellowship training in neurology at Johns Hopkins University. A common theme to his research has been in the study of the astrocyte biology and its role in disease pathogenesis, with a particular emphasis in how astrocytes may contribute to ALS. Dr. Maragakis has been heavily involved in clinical research as the principal investigator, site principal investigator, or co-investigator of numerous clinical trials in ALS. Many of these trials were coordinated by the Northeast ALS Consortium, on which he has served as an executive board member and is currently a member of their scientific advisory board. Dr. Maragakis has received several awards for his outstanding research efforts. He is a member of the American Board of Psychiatry and Neurology, the American Academy of Neurology, and the Northeast ALS Consortium. I will now hand it over to Dr. Maragakis for his presentation.
Great, thank you very much. And thank you to the ALS Association for allowing me to share some of the work from the scientific community as we try to think about how to translate science into ALS clinical trials. These are my disclosures: consultation, advisory boards, research support, and clinical trial research support. I'm just not gonna spend a lot of time on this, but ALS often is described as a rare disease, but in fact, it's an uncommon disease, but the most common motor neuron disease, with an annual incidence of 2 to 3 per 100,000 people, and an average age of onset about 55 years of age, although certainly in my clinic and other clinics like it, we see a very broad range of patient ages. The prevalence of ALS seems to be increasing over time, and whether that's due to the aging population is not quite known. One of the biggest challenges we have currently is that the survival, of course, after disease onset or symptom onset is 2 to 4 years, but an important subset of patients, maybe 5 to 10%, live greater than 10 years, and that's an important subset for helping us understand what may be different among these patients. So ALS is really a disease characterized by the degeneration of what we call upper motor neurons, that is the neurons of the cortex or the brain, often called Betz cells. And then the degeneration of the descending pathway is called the corticobulbar, that is the tract that go to the speech and swallowing, and the corticospinal tract. And that's important, as I'll touch on these pathways later. It also involves the degeneration of what we call lower motor neurons. It is the neurons that go from the spinal cord out to the muscle. And it's this combination of nerve degeneration over time that leads us to a diagnosis of ALS. Importantly for this talk is that although 90% of ALS or people living with ALS have what we consider sporadic disease. An important subset, about 10%, have hereditary ALS, where it runs in families, and there have been genes that have been associated with these familial forms. And I want to highlight a couple of them. One is called the SOD1 mutation, the first ALS mutation to be discovered. But the most common one is called C9orf72, and that's probably present in about 7 to 15% of all ALS patients. There are now over 30 genes that have been linked in one way or another to ALS. Importantly, about 15% of ALS patients have the coexistence of dementia, or specifically frontotemporal dementia, which has really broadened our spectrum of what we consider part of ALS, rather than just being a neuromuscular disease, as part of a general neurodegenerative disease. So keep these genes in mind as we'll revisit them later. I put this slide up not to go through the details, but the first thing that always strikes me, and I hope may strike you as well, is these are all different pathways and different genes that are thought to be relevant to ALS pathogenesis. And you can see all of these different pathways, including on the far left, neuroinflammation or abnormalities of mitochondria, transport called nucleocytoplasmic transport, glial dysfunction, we'll talk a little bit about that, excitotoxicity, and so forth. So many of these pathways are thought to be involved in ALS. And I think that's important because ALS is probably not one disease, but probably represents a spectrum of disease with a common final challenge, which involves motor neuron loss. And I think that's important as we move forward from science to the clinic, because what we hope is that we will be able to target these individual pathways to treat ALS patients as individuals rather than just as a large group. The other thing I want to stress is ALS is more than a motor neuron disease. And through our work and the work of many others, we know that it's just not these motor neurons in the middle that are affected, but other cell types seem to be involved in the way ALS progresses over time. And these include things like astrocytes, here in blue on the left, microglia, these little inflammatory cells that seem to be like kindling to a fire to really turn on motor neuron loss, and even things we associate with multiple sclerosis like oligodendrocytes. So all these different cell types are playing a role in how the disease changes after it starts. And why is that important? It's important because each of these cell types can serve as potential targets for ALS therapeutics. So I've shown you these kind of complicated slides. This could be confusing, but where do we start? Where do we start? We're going to start with a particular protein that really is currently thought to be one of the nidus for ALS, and in fact, other diseases like Alzheimer's disease as well. And the protein is called TDP43. And TDP43 was called by one of my colleagues the guardian of the transcriptome. You maybe heard of guardian of the galaxy. This protein is the guardian of the transcriptome, right? And the transcriptome is a way to think about it is this. If you've ever played here in the bottom right-hand corner, the game telephone, where someone whispers a sentence and then it's passed on and on. And by the end, oftentimes that sentence is quite convoluted. Well, if we were to use as an example, the starting sentence, I am not going to the store today. The TDP43 is responsible for actually making that sentence turn out the right way. So if my intention is to actually go to the store, the TDP43 clips out or oversees the word not. So instead of I am not going to the store, that sentence should be I am going to the store. But you could see if that word not is left in there, the meaning is completely confused. And that's much like what happens in the cell. The messages are not transcribed appropriately, and things start to fall apart. So what goes wrong with TDP43? So it's called an RNA binding protein, and it regulates the role of something called RNA splicing. That's kind of fancy scientific terms. But here in this picture, I want to show you that you see the blue. The blue is what we call the nucleus of the cell, the center of the cell. And the brown and the blue should really overlap, like you can see with this asterisk. But a few of these cells, there's brown stain, this TDP43, that's sitting outside of the nucleus in something called the cytoplasm. It's not where it should be. And so when this TDP43 is outside the nucleus, it can't carry out its function as a guardian of the transcriptome. It can't get to where it's supposed to be. And that leads to the aggregation of proteins. It leads ultimately to cell death. And in fact, this TDP-43 pathology that you're seeing in this picture is present in over 90% of cases or of individuals with ALS. Okay, over 90% of individuals with ALS, suggesting it's an important protein that we should pay attention to. So if this protein is here in the middle, and it's supposed to be in the nucleus, and it's supposed to shuttle between the nucleus and what we call the cytoplasm, what happens when it can't get into the nucleus or stay in the nucleus? Here in red, I've circled the what we call aberrant RNA metabolism. What I was telling you about the message, I am going to the store, I am not going to the store. This message doesn't get appropriately transcribed, and so things start to go wrong downstream. You can see that TDP-43 has a lot, you see a lot of these arrows, has a lot of abnormalities that gum up the cell. So there's more than one problem or more than one function related to TDP-43. But I'm going to highlight what goes on with the message in today's lecture, because I think not only is it important scientifically, but I'm going to show you how it's relevant to how we're thinking about clinical trials. So when I go to the clinic, what are some of the things that I think about? As a general rule, why does weakness in ALS seem to have a distinct anatomical spread? That is, if a patient presents with foot weakness on the right, I can tell them that it's likely that their right foot or right leg will continue to get weak before they would get, let's say, left hand weakness. Why do some ALS patients progress more slowly over years or even decades and others more rapidly over months? Why do some patients present with what we call upper motor neuron disease and others with predominantly lower motor neuron disease? Why do those patients look so different? Why do some ALS patients have bulbar onset ALS with speech and swallowing problems, while others have spinal onset ALS, like trouble with gripping or trouble with walking? Why are certain cell populations spared? For example, eye movements are spared. Autonomic function like bowel and bladder and heart function are spared. And importantly, ALS is no longer thought of as just a neuromuscular disease, as I mentioned before, but a neurodegenerative disease with associations with disorders like frontotemporal dementia. So if that's the case, these are some of the questions. How do I and my colleagues start in the laboratory? How do we model ALS? And this is a mouse model of ALS. And you can see, I hope in this picture, that this mouse develops weakness of the hind limbs here in the back, eventually weakness of the forelimbs, and then develops respiratory failure and trouble chewing and swallowing, much like we see in some people living with ALS. So what are some lessons from this mouse model of ALS? Number one, the way this mouse model was made was genetically, and it was made through a mutation in the SOD1 gene, the one I told you that represents about 1% of all ALS patients. So this mouse only represents a very small fraction of all the patients who have ALS. So that's a limitation of this mouse model. This mouse model has extremely fast decline in limb strength and survival, so may not give a chance for drugs or therapies to have a good opportunity to take an effect, because this occurs so rapidly. However, the mouse is useful for understanding drug toxicity. What if side effects might a drug of interest or a clinical trial drug of interest have on the liver or have on the brain? It's useful for understanding what we call the blood-brain barrier. If I give a drug through the veins or by mouth, can it get to the brain? Can it get to where it belongs? And we can't do that in a dish. We need a mouse model for that. And it's also useful for understanding some pathways relevant to ALS. So what I'll tell you is in the 90s and early 2000s, this mouse model was used as what we call the go, no-go decision. If your drug worked in this mouse, it went forward to being tried in people living with ALS. Now we don't necessarily feel that way. So my message to you is that we still do use this mouse, but we're learning a lot about asking the right questions and using the mouse more strategically to think about how to move drugs forward, okay? So how to move drugs forward, because it probably only represents about 1% of all patients. Encouragingly, there are now a variety of other models that I and my colleagues can use in our laboratory. I mentioned mice. There are worm models of ALS called C. elegans. This is these green worms are seen under a microscope. Drosophila, this looks like a scary picture, but this is actually a fruit fly. And fruit flies can be great for manipulating genes related to ALS. Zebrafish are good models that we can study ALS. Cell culture, and I'm going to talk to you a lot about cell culture today. And finally, even yeast have been used very elegantly to find genes that may be relevant as we think about ALS therapeutics. So a lot of tools now at our disposal that we're trying to use. So I mentioned earlier, there's a lot of what we call heterogeneity. Every ALS patient looks a little bit different, how they change over time, where their weakness might be. So if there's such what we call heterogeneity in ALS, are all mechanisms of neuron or motor neuron degeneration similar? And I think the answer is probably no. So if that's the case, are there opportunities to use human, what we call iPS cells or induced pluripotent stem cells to address this ALS heterogeneity? And why are these stem cells useful? Induced pluripotent stem cells, really discovered in 2007 and 2008 by Yamanaka and colleagues, for which he won the Nobel Prize for science and medicine, allow us to take, let's say in the case of people living with ALS, their blood or their skin cells, reprogram those cells into what we call induced pluripotent stem cells that I think you could see here. And then we can make them into the cells of interest. In our case, we're very interested in motor neurons, right? So we can do that from patients. In fact, potentially, some of you may have participated in studies where your blood was collected and your blood was made into stem cells through programs like Answer ALS and others. So we can make stem cell types of interest or cell types of interest like astrocytes that I mentioned earlier, motor neurons, microglia, and we can put those in a dish and we can mix them up and ask what kinds of relationships these different cell types have. We can also make them into very specific subtypes. I mentioned earlier that there are upper motor neurons called corticospinal neurons. We and others are making iPS cells into corticospinal neurons. We can make them into spinal motor neurons as well. And then we can put them in a dish. And in fact, we can even record electrically from those individual cells under a microscope. So these are all ways that these induced pluripotent stem cells, representing hundreds, if not thousands of patients, can be used very individually, something we call a precision medicine approach. So how do we use these stem cells? We can ask questions about ALS-specific pathology. Are there specific abnormalities in one individual's cells that are not present in others? We can record electrically from these by putting an electrode into these cell types. We can stress the cells. Patients oftentimes ask me if stress or other things can lead to cell death, and I think the answer is yes. We can model cell-to-cell interactions, and we can even, importantly, screen drugs in a dish. And I'm going to show you some examples of that. Again, in my lab, in the laboratory of others, we can make them into very specific subtypes. We can turn them into spinal cord neurons or spinal cord astrocytes or cortical neurons and cortical astrocytes. And we can, and a lot of other cell types for that matter. So our tools available for modeling ALS have really grown really within the last decade. And this is just an example from my laboratory where we're making induced pluripotent stem cells into brain cells and into spinal cord cells, and we're putting them in two different chambers. And we're allowing the nerve cells from one to grow through some tubes into the other. And they can actually connect electrically. And we can actually stimulate them with light and show electrically. You can see the responses. These are electrode responses in one compartment here on the top, which is here. And then the response in a lower compartment down here, which is right here. And we can show that, in fact, those cells are connected. And again, this is an hour talk in and of itself. But even at the microscopic level, we can see this is an individual nerve cell fiber that we can see that those cells are talking to each other. So that's another way of modeling ALS. Now, how about genetic studies? Really important as we think about ALS therapeutics. This goes up to about 2020, this particular graph. But here in the red circle was the first gene. It was called the SOD1 gene, discovered in 1994. The mouse model that I showed you was described in 1995. But you can see the tremendous rise in the number of genes that have been discovered related to ALS. And this now gives us a lot of tools, but I'm going to focus on this particular gene called SOD1 because we have new data in the last couple of years that have led to the approval of a drug called tofersen, or maybe Qalsody. So what is that drug called tofersen or Qalsody? It is an antisense oligonucleotide. And what is an antisense oligonucleotide? It's a little piece of DNA that binds to the abnormal gene. It binds to the abnormal gene, the abnormal SOD1 gene, and it calls on some other parts of the cell to destroy that protein. Okay, so destroying the protein of interest. And this can be designed for any number of genes. It doesn't have to be related to ALS. It could be any gene in the body. So these are designed to be very targeted. And again, the key word here is targeted just to that gene. There's a lot of fancy biology here, but what I want my audience to know is this is now a strategy tying genetics to very specific drug therapies. Now, unfortunately, these antisense oligonucleotides for ALS or for other brain diseases can't cross into what we call the blood-brain barrier. So you can't give it into the vein or take it by mouth. It has to be given via a spinal tap, right into the spinal fluid. So this requires, in the case of this drug called tofersen, or known commercially as Qalsody, monthly lumbar punctures right into the spinal fluid. We've had patients now who've been doing it for years. It's well tolerated, but it's certainly not as easy as taking a pill. And we'd like to be able to get there eventually. But for now, this is how we get it into the brain and spinal cord. So this was a tremendous breakthrough. I also want to emphasize that this doesn't happen overnight. Drug discovery and drug successes and failures, for that matter, oftentimes take decades. So to highlight that, I just showed you that gene was discovered in 1994. And it was about 2023, I think, when that drug was finally FDA approved for ALS patients. So a number of years. However, that shouldn't be discouraging because as we've discovered more genes and we've discovered more about antisense oligonucleotides, that process speeds up. And we've seen that with other neurological diseases where the rate of discovery is accelerated as we learn more about drugs and we learn more about genes and we learn more about the science leading to these clinical studies. So how does this antisense oligonucleotide work? And this is a lot of fancy biology, but very simply here in red, this drug targets the abnormal gene or the abnormal protein, and it also, interestingly, just targets the normal protein. So you don't want to turn down the protein to zero. It knocks about half of the protein down. And it turns out, and that was a very important part of the original studies, for safety. We didn't know if you knock that protein down, would it be safe? And that's why as we develop drugs, we do what we call different phases of studies. And the first phase, this you see here, this was a New England Journal paper called Phase 1/2 study of antisense oligonucleotide tofersen for SOD1 ALS. So the first part of that study in 2020 said, this drug is safe. It was followed by this really groundbreaking study that followed a couple of years later, suggesting that the drug did in fact have an effect. And this is important. So this is the interface between science and clinical trials. So this is called a biomarker. The first question we wanted to know is, in ALS patients, did this drug do its job? Did this drug do its job? And in fact, the answer was yes. Here in blue, this is time along what we call the x-axis. And here in blue were patients in the first 6 months of the trial who got placebo. And in red were patients who got the drug tofersen. What you can see is the patients who got the drug had a lowering of the SOD1 levels. And then at 6 months, everyone got drug. And you can see that after 6 months, those levels of SOD1 went down, right? So that's really important. It met its target. It engaged its target. Neurofilament light, called NFL, not National Football League, but neurofilament light, is a protein in nerve cells that gets released into the blood and into the spinal fluid when nerve cells die. And it's currently probably the most reliable marker for ALS and other neurodegenerative diseases. So similarly, patients who got the drug, those neurofilament levels went down in the first 6 months, and patients who were getting placebo had no effect on that, what we call biomarker. But then after they began getting drug, their levels went down as well. Importantly, there was also a trend in this early study to suggest that patients who received drug had a slower disease progression, their breathing function was relatively preserved, their strength was relatively preserved, and survival was relatively preserved. So although this study did not achieve its what we call statistical significance for slowing disease course, the trends that I just showed you were very encouraging. So much so that it led to the FDA's approval of this drug, like I say, I think back in 2023 now. More recently, in fact, this month, a study with this same compound in patients, now this is, in this case, 7 patients. I'm showing you these neurofilament levels. And in 7 patients who are now out, you can see 30 months, so that's almost 3 years with the disease. Giving patients that drug really reduced the levels of neurofilament and one called neurofilament heavy. So again, it met its target. It did what it was supposed to do. And encouragingly, whoops, you can see here that there's a suggestion that patients who were taking this drug had a relative plateau in their ALS functional rating scale, a plateau or potentially even an increase in their muscle strength. An increase, this is patients at baseline and patients more recently, had an improvement in what they call the FIM motor score, an improvement in their 10-minute walk test, and in a variety of tests like we call something called sit to stand. So this is really potentially interesting because it could suggest for the first time that not only ALS could be slowed or potentially even stopped, but patients have the capacity for potentially for improvement. So that's really exciting. So how do we get there? I'm going to shift gears a little bit because this is important in this interface between science and clinical trials. And that is a push for biomarkers for ALS. What is a biomarker? A biomarker tells us something about disease. It gives us something to target. I'll give you an example. In the cancer field, we know that a cancer drug works when we take a CAT scan or an MRI scan and we can see that the tumor shrinks. Well, we don't have very good measures in ALS, let's say, if MRIs aren't great for seeing differences in ALS. So are there potential biomarkers? Well, we can get biomarkers from a lot of different things, from blood, from brain and spinal cord tissue, from urine, from skin, muscle. We can even look at electrical measures of ALS as biomarkers. PET imaging, something called positron emission tomography, and we're doing that here in our laboratories. We can get spinal fluid from ALS patients. And so there are actually some very important studies, and you're going to hear about, I think, later in this week about all ALS. That's a nationwide effort to look at biomarkers related to ALS as part of the ALS community. So biomarkers are critical components for design of ALS therapeutics. They can tell us something about diagnosis. That is, do I have ALS or do I not? It can tell us something about prognosis. If I have a high level of a biomarker, it may suggest that I have more rapidly progressing disease. And if I have a lower level of that biomarker, maybe I have a slower disease progression. A predictive biomarker might be something that we would use in a clinical trial. That is, patients who have biomarker X might be a great candidate for this drug, whereas patients who have biomarker Y might be better suited to another drug. And finally, pharmacodynamic biomarkers. And that are biomarkers just like the NFL or neurofilament light that I showed you that tell us that our drug is doing what it's supposed to be doing. We don't want to try a drug that we don't think is getting to the right target. Okay, so those are pharmacodynamic markers. So let's revisit this picture. This TDP-43 is a linchpin or nidus for disease. How might we combine some of these efforts? So I showed you these pathways that we're talking about. So the first question is, can TDP-43 be used as a biomarker for ALS and this FTD, frontotemporal dementia? And the answer is not yet, but hopefully you can see here on the left all of these different studies with ALS patients looking at TDP-43 as a potential biomarker, and things like spinal fluid and things like plasma, blood, and so forth. So I think hopefully the time will come when we can use TDP-43 to tell us more about how patients are changing. In the meantime, an important part of TDP-43 is what it can do downstream. And I told you earlier about it being the guardian of the transcriptome, keeping that message on message. And so I want to show you here, because this is important for the rest of what I want to say. This here in orange is TDP-43, this protein. And what it normally does is take something called a cryptic exon. That is part of the message. It's not supposed to be there. It takes it out. Just like the word I am not going to the store becomes I am going to the store, right? Let's take that word not out of the equation. So normally that's what TDP-43 does. If it gets messed up, that cryptic exon stays in there and it ends up saying I am not going to the store, causing confusion in the cell. And that protein gets either does things it shouldn't be doing or gets gums up the system, if you will, right? So that's what happens with TDP-43. It's not where it's supposed to be. It's not doing its job. And so it wreaks havoc. So what does it wreak havoc on? What proteins does it wreak havoc? So this was an important study by a colleague here at Hopkins named Phil Wong. And he looked and said, what are those messages that TDP-43 is missing in ALS patients? And he focused on a number of these different, what we call cryptic exons. And he found that in patients with the C9orf72 form of ALS, and we talked about that being one of the genes related to ALS, that even pre-symptomatically in patients who carry this gene, or symptomatically, that certain abnormal messages were higher than they should be. Okay, and if you focus here on this red square, you see that his group looked at patients, what we call normal controls, so patients who don't have ALS, compared to the ones in red who did have ALS, even before disease starts. Even before disease starts, the levels of these abnormal messages or cryptic exons were higher than they should be. Okay, and he went on to look, and he looked at one of these called HDGF L2. It doesn't matter what the name is. There are a lot of these. And he found that even before disease started, these are patients who carry a gene for ALS, but don't yet have ALS. This cryptic exon, this abnormal message, was very high. And then over time, he thinks as cells die, this level came back down. But what did go up was neurofilament, this same protein I told you earlier, which is a sign of ALS. So where we want to be as a community in clinical trials is way back here, before disease even starts. So before disease even starts. And that's where the field is going. That's where the field is going with regard to SOD1. That's where the field is going with a lot of these genetic disorders. We want to be able to identify patients and help them before they ever get symptoms. And this was reproduced by our colleague Michael Ward at NIH, and he found a whole list of these, what we call cryptic exons, abnormal messages, a whole list of them here. And he looked in 15 different patients here, and everything you see in blue are the abnormal accumulations of some of these abnormal messages. And you see here that some patients have different abnormalities. So not every patient looked the same. And that's important as we think downstream. You can imagine a day where patient number 1 gets a different drug than patient number 6. Okay, so they look different. So this gets to the idea of precision medicine. So are there therapeutic opportunities for restoring TDP-43 function? We hope the answer is yes. And a colleague is using a gene therapy, or he hopes to use a gene therapy, something called AAV9. It's used in clinical trials for other disorders. And he's combined it with something called CTR, chimeric TDP-43 Raver. Fancy name, but the idea is that this gene would get into a nerve cell and restore the function of TDP-43. And as he points out, TDP-43 loss of function occurs in about 97% of ALS and about 50% of Alzheimer's disease. So it's a novel gene therapy that rescues the loss of TDP-43. Importantly, this AAV9 CTR can cross the blood-brain barrier. So ideally, it can be given intravenously and get into the brain, get into the spinal cord where it needs to do its job. It's already been demonstrated what we call in vivo, that means in living animal models, that it works. And proof of concept studies have been performed in mouse, fruit fly, and cell culture. And here's one example. This is a fruit fly model. And I'm not going to go over all the fundamental science here, but I just want to draw your attention to the lower left corner. This is the survival of a fruit fly in days. So this is the percent of fruit flies that are living, and this is the number of days they're living. So you can see that an average fruit fly, let's say, lives about 50% of them live about 50 days. And if they have abnormalities of this TDP-43 protein, they live for about 5 days. And if he gives these fruit flies some of these AAV9 gene therapies, he can get them to live longer. And similarly, when he looked in a TDP-43 mouse, that is a mouse that harbors abnormalities in this TDP-43 protein, they normally decline in body weight over time. He can show that giving them this drug preserves body weight, preserves their ability to run on a rotating rod. They have a prolonged ability to hang on a wire, and the disease onset, that is their ability before they develop disease, is also prolonged. So two important studies suggesting the capability of this particular approach. What are some of the other targets of TDP-43? So UNC13A is a protein. There's a lot here. So luckily we're recording this because you can always go back. But UNC13A is a protein that's important in what we call the synapse. And that is the point at which one nerve cell talks to another nerve cell. So one nerve cell signals another nerve cell. And this particular protein is important in synthesizing that message. So I am not going to the store, I am going to the store. It's part of that, method of communication. And when TDP-43 doesn't work right, that protein called UNC13A gets messed up. And so the signal from one cell to another gets messed up. And so there have been some beautiful models in cell culture and other models to suggest that this target is relevant. It's linked to TDP-43. And oh, by the way, there are some potential treatments or some potential drugs that could be tried, like lithium carbonate. And from a company called Curalis, they've developed, again, if we revisit this antisense oligonucleotide that we revisited for SOD1 protein, to maybe target UNC13A missplicing. So again, now there's an ASO, an antisense oligonucleotide, targeting a brand new gene, a brand new protein. So again, another relevant target. Similarly, Stathmin-2, a different target. Stathmin-2 was found in cell culture models, again, science to bedside, to be abnormal in ALS patients and modulated by TDP-43. And Stathmin-2 is an important protein that allows nerve cells to grow out into, to communicate to other nerve cells. So its ability to grow out what we call axons, or grow out little nerve bundles. And so when this Stathmin-2 protein isn't working correctly, the ability to regenerate or to maintain that nerve projection is lost. So ANQUR is a multi-center, double-blind study going on in Europe as well as Canada from, again, a company called Curalis. And it is also a drug that rescues this Stathmin-2 expression. So it's another ALS relevant target, again, linked to TDP-43, but again, you can see that this is a different target. This is involved in axon sprouting. The other one is involved in nerve cell communication. So again, two different targets. So hopefully I've been able to show you how what we do at the bench, identifying very relevant genes, can lead to potential drug targets and drug designs. So in the last couple of minutes, what are some new ideas in clinical trial design? I want to emphasize that revisit the fact that there are a lot of different ALS targets that I think are really relevant. I just highlighted a few, right? This is just a handful of the many ALS targets that are out there. So this, hopefully you can see that there are nerve cells in red, there are astrocytes in orange, microglia in light orange. All of these are relevant cell types involved with relevant ALS therapeutic targets. So there's a lot going on. How has clinical trial design changed to afford better participant experiences and outcomes? How do people living with ALS, how have they changed how we as scientists and clinicians think about it? Well, technological advances like remote monitoring of clinical trials using telemedicine. We learned a lot during COVID, right? We learned a lot during COVID about telemedicine. Use of wearables, use of the cell phone to analyze patient speech and mobility, et cetera. Artificial intelligence and big data analytics. I'm not an expert in this, but this is coming. Personalized medicine, I mentioned, genomics and biomarkers. Globalization of trials. I mentioned studies going on in Europe, Australia, North America, South America, et cetera. Patient-centric approaches. How can we make clinical trials easy for people living with ALS? And then public-private partnerships with organizations like the ALS Association, those relationships to help make clinical trials more accessible. How might clinical trials change in 2025? I think you're going to hear a little bit about this week, so I won't belabor it. But my guess is clinical trials will go on a little bit longer. Rather than 6-month trials, probably 9-month trials or 12-month trials to get us a better sense for drug treatment. Probably shorter time from symptom onset. Instead of 3 years or 4 years from a symptom onset, maybe 18 months or 2 years. An emphasis on biomarkers like neurofilament, NFL, which are now being used to decide how or should we move forward with a particular drug of interest. And then validation of target engagement. Did the drug provide the expected effect on its target? And then this last slide, could artificial intelligence allow us to predict which drugs might be more successful in ALS? There are now a lot of efforts going on. This is just one publication, and I will not at all pretend to understand how artificial intelligence can do that, but I will tell you it's really exciting from what I'm hearing from experts in the field. So what are future directions? Developing better animal models and better cell models of ALS that can aid in the study of novel therapeutics. Invest in the development of better biomarkers for understanding disease progression. Define the clinical and genetic subtypes of ALS to allow for stratification of patients into the most appropriate clinical trials. And finally, continue to expand the therapeutic landscape. So those are some of the things where I see the future going. So I'll stop there. Hopefully I've left a little time for questions. Again, we could have spent a day really talking about all the different drug trials that are out there, but hopefully what I've shown you today is how my colleagues and I get from the bench to hopefully getting drugs to patients. Okay, so there's a lot here. Okay, so
yes, thank you, Dr. Markakis. Do you want some help, or can you see them?
I think I've got it. No, thank you. That's a good one. I'm going to start. I'll try to get to as many as I can.
Okay.
Wow, they're moving fast. Okay. One says, you've detailed much about genetic ALS, but how does this relate to 90% of ALS patients? They're a single marker for all patients. So I want to highlight that the answer is yes, and I think that's what's so exciting about TDP-43. I think TDP-43 is important, we believe as a community. It's important for 97% of all ALS. So I think that's something we can hang our hat on. I showed you a biomarker called NFL, which is also present in all ALS, high levels, and we can actually look for that in the clinic. We can send a test away for high levels of NFL. So I think the answer, and I didn't even detail a lot of the other targets, like targets for neuroinflammation or targets for immune-mediated therapies. or mitochondrial abnormalities, et cetera, probably apply to all patients with ALS, not just those genetic forms. Um, so hopefully that answers a little bit. Assuming any of the ALS therapeutics treatments prove successful, would the gains restoring motor neuron function be less for people who have progressed more rapidly? Um, I guess the answer is a little hard to know. In other words, can we restore function for patients who have progressed more quickly? I think the hope is to be able to, and that gets into the question about diagnosing patients earlier, identifying patients even before they may have disease. And that goes, I'll give you an example. We use a PSA from our primary care doctor to diagnose or to address prostate cancer before it starts, right? Before it starts. So is there something where you first see your neurologist with just a little foot weakness that would allow us to identify patients with ALS before their symptoms became severe? So I think the answer is the study I showed you suggests that we can halt progression and potentially even restore function. I think the more rapid the decline, the more challenging that becomes. But again, I think these are early days. Riluzole is a common medication used with ALS. How does it work? Well, riluzole probably works in a few different ways, but one of the ways in which riluzole works, by the way, I will say it works reliably. It's been shown over the last 30 years to work reliably and reproducibly. It's not a home run, but I think it does form the cornerstone of therapy. It works by when a cell gets irritated or it dies, it tends to release a neurotransmitter called glutamate. We all have glutamate in our brains, but when too much glutamate gets spilled into the surrounding brain and spinal cord, it overloads cells and it tends to overexcite motor nerve cells or motor neurons, and those cells subsequently die. So it essentially reduces the release of that glutamate. And so it's, again, a very reliable target that's been reproducible over time. Aside from saliva tests, what other ways can specific gene mutations be identified for sporadic ALS? Well, I assume what you're referring to is, at least in our clinic and many others, to do gene testing, we do a mouth swab that collects saliva and some of the surrounding skin cells of the mouth. There are other ways. Any way you can get a cell, so that blood, skin, those are ways that genes can be analyzed. Saliva turns out to be easy. It doesn't hurt. It's easy to manage. So there are lots of other ways. Urine, all those can be analyzed, but and there are currently studies to try to do that. But it turns out saliva for most gene testing is pretty easy. Let me see what else we have. Are you looking specifically at slow progressing ALS to help guide study design and therapeutics? Great question. It's got a lot of layers. So one of the challenges with slow ALS for clinical trials is it's sometimes hard to predict how fast patients might change, right? And so it's hard to study a drug in a slow progressing patient who might have had disease for 5 years or 7 years or 10 years. Should they be taking a drug for that long? My belief is that the answer is yes, there is a role for patients with slow progressing disease. And the idea could be, and I've just shown you some data today to suggest for patients with slow progressing disease, maybe those are the patients who might actually benefit from getting some recovery. And I think that's why that one of the studies I showed you is potentially exciting. So most trials have slotted patients into a certain time frame, but I think there is a potential role for some specific therapies for patients with more slowly progressing disease. Are there any trials for PB? I assume you mean pseudobulbar palsy. Are there any trials for pseudobulbar palsy? Well, there are some symptomatic drugs for pseudobulbar palsy. You've probably heard of Nuedexta, which turns out to be a very effective drug for symptomatic forms of pseudobulbar palsy. We don't necessarily understand what makes pseudobulbar palsy. My belief is it's a form of ALS, but what are there any unique molecular markers that make it different from ALS? I don't think we have a good handle on that yet. One example, it's not quite pseudobulbar palsy, but bulbar ALS, was the drug prodopidine, which was studied as part of the Healy platform trial, which did show that that drug may have had more benefit in patients with bulbar symptoms than in patients with what we call spinal onset ALS. So it gets a little more to your question. Whether that drug will be more effective in patients with bulbar onset ALS remains to be seen. So yes, symptomatic therapies that exist for pseudobulbar palsy, but I think many of the drugs that could be relevant for ALS will also be relevant for pseudobulbar palsy.
Dr. Maragakis, I'll just hop in because I see a couple questions that we can maybe combine
Sure.
that you might be able to answer, you might not. One that I saw a couple times was just how the new administration and withdrawal from the WHO might affect ALS research, and if that will affect ALS research or the development of drugs for use in the US. I know that's a tough one. So,
Well, well, it's quite frankly very straightforward. So laboratories like mine and my colleagues around the US are reliant on the National Institutes of Health. We get federal funding for grants. I have an NIH grant that drives research. And so the answer is yes, that it could have an effect if if funds are delayed or withheld. So regardless of the cause or whatever that downstream might happen, that's how most of us get the majority of our funding is through federal grants, through the NIH in particular.
and if you'd like, I can read a couple more that were maybe posted a little earlier in the chat. Is there a way to get your TDP 43 level tested?
So the answer is, to my knowledge, is currently no. And it's not so much about the challenging thing about TDP 43 is it's probably not about the level of TDP 43. It's probably about where TDP 43 is in the cell. So it doesn't necessarily have to be high or low, but probably the biology is probably a little bit more complicated than that. And that is where it sits in the cell and whether it gets, whether it gums up the system. So the outside of research, the most reliable biomarker for ALS that's commercially available, let's say where your neurologist could test, is NFL, is neurofilament, and that can be sent away to a clinical laboratory and can give you a range of high or low, or high or normal or low. And we do know that that will tell us a little bit about disease progression. Does exercise affect the speed of disease progression? We get this a lot. So much so that we did a trial here at Hopkins of 3 different forms of exercise. We looked at stretching and range of motion, which I think is important to do every day. And then we looked at 2 different additional forms of exercise: endurance exercise in the form of pedaling a stationary bicycle, or resistance exercise in the form of light weights. It was a study that was actually sponsored a few years ago by the ALS Association. What we found was that exercise did not cause disease to speed up. It was found to be safe and well tolerated, and there was a trend towards patients who had certain forms of exercise potentially to have fewer falls. So, practically speaking, what I tell my patients is they should be doing everyday stretching and range of motion exercises. And then on a practical perspective, I ask them to do maybe 3 days a week, a Monday, Wednesday, Friday regimen of something a little bit more rigorous. How much is too much? I tell patients you shouldn't be fatigued, sore, lying on the couch the next day. That might suggest that you've overdone it.
Okay. One that might be easier for you to answer. That was a little earlier in our Q&A. Do you have any interesting clinical trials coming up at Johns Hopkins, and how could people get connected with those trials?
I would say two things. We're very privileged to be part of every academic site, including Hopkins, will run some clinical trials that are unique to Johns Hopkins. And I didn't detail some of those, but we have some that are maybe smaller that we're running only at our site. And if you'd like access to that, you can go to our website, the ALS Clinical Trials Unit at Johns Hopkins. So Johns Hopkins ALS Clinical Trials Unit, that'll give you a number to contact. And the second part is we're very privileged to be part of a national organization, in fact, a global organization called NEALS, the Northeast ALS Consortium. And that really has allowed places like Hopkins and others to be part of a much larger network of patients. I will say, this holds true for me and probably my colleagues as well. If you're seen in my clinic and a particular study is full, I'm happy to send you to one of my colleagues to participate in a clinical trial in Philadelphia or Washington, D.C. And I think most of my colleagues feel that way. If you're in, New York and you don't have an opportunity for a clinical trial, you can still be followed in New York, but be seen at Hopkins. So I like to think it's a really good community in that respect. So yes, we have a website and contact information.
Okay.
I will say, by the way, as a side, there are trials going on now, and there are some really exciting trials actually coming up in the next quarter, I would say.
Great. Ryan, should I read a couple more questions, do you think?
Sure.
Yeah. Okay. Let's see. What have we got? Let's go from the bottom down. Have they determined that a cause of ALS is exposure to toxic substances? The reason I ask is that I saw that the Federal Agency for Toxic Substances and Disease Registry established the National ALS Registry in 2010 to collect and analyze data about people living with ALS.
And
Yeah,
That's a tricky one, right?
I just wanted to hop on real quick before Dr. Maragakis discusses that. Just to mention, we are going to be diving into the CDC ALS National Registry on Thursday at 3 p.m. in that webinar as well. But I'll allow Dr. Maragakis to answer.
sure. I'll say I think the answer for the most part is we don't know. And I would say part of that is there's no one there's no single toxin, I would say, currently that we can hang our hat on. Does that mean that toxins can't play a role in ALS? I would say we just don't know. I would say the important thing is that there are colleagues around the country who are doing a very nice job of collecting some of those data with regard to toxic exposures and so forth that may be relevant. And by the way, it may be that this is kind of I'm sure it's a cop-out, but not everyone may react the same to a toxic exposure. So it might be that you need a little, what we call a genetic predisposition to react to a toxic substance. But it's not out of the realm. We know for disorders like Parkinson's disease that there are toxic substances that can predispose to disorders like Parkinson's disease.
Thank you. Let's see, how many, oh, no. Do you believe ALS clinical trials can be conducted quicker and less expensively outside of the United States?
It's a good question. I'm not sure about, so I don't think more quickly with regard to once a patient is enrolled. I think as a community, we've kind of come to the conclusion that if we get better biomarkers, we could shorten the time of a clinical trial. But I think to see how a patient responds, we're probably still talking about months a duration. Can they be run elsewhere more quickly? Potentially for large patient populations, right? So, East Asia certainly would be patients with millions and millions of patients. But we just haven't really seen that come to fruition. So I would have to say that Australia, North America, Europe have done the bulk of clinical trials, at least with regard to getting drugs to patients. But that doesn't mean it couldn't be done couldn't be done elsewhere. I will also say, by the way, that patients may respond differently. Oh, by the way, I should include Japan as well. Patients may respond differently depending on those patient populations. So I'll give you an example. In East Asia, the C9orf72 mutation is extremely uncommon. In Scandinavia, the C9orf72 mutation makes up the majority of familial ALS patients. So it may be what's good in one population may not necessarily translate to another. So it's something to keep in mind.
Great. I guess I'll segue that into, Gabrielle had a question. How many genes should I expect to get tested for ALS nowadays?
So we send with a couple of different companies that I think it's above 35. I think it's between 35 and 40, but I don't know the exact number. And what I would say is this: there are a handful that are the most common, and then some of them are, much less common as we go down the list. My anticipation is we'll discover more genes, and we'll discover genes that are make patients at risk, but don't necessarily directly cause disease.
Great, thank you. I think we have time for one more quick question. How to choose. Are you optimistic about MN166?
So that's from a company called Medicine Nova. The drug is called Ibudilast.
Mhm.
It's been through a number of different phases, showing safety, showing a rationale behind why it should work. So I would say this: I tell patients, at least here at Hopkins, we're doing that study. We wouldn't do any study we did not consider scientifically sound. Do I know so I'm optimistic about any of the studies we do. I think they all have a good rationale. So I think the answer is yes, and I think we will stay tuned.
Thank you. Thank you so much, Dr. Margakis. Thank you for that was a wonderful presentation. Unfortunately, we're out of time. So if you weren't able to get your question answered, you can send it to Ryan, and he'll work to get you an answer. His email is Ryan, RYAN, .tappe@als.org. And thank you for just all the information and just all of your support, Dr. Margakis. It's wonderful. We hope those in attendance today found the information helpful. Please join us again tomorrow at 3 p.m. for our third webinar of the week. They'll be discussing the Access for All in ALS Consortium. And a final reminder that a survey should pop up on your browser when we end the webinar today. Please don't forget to complete that survey. It really helps us a lot. Thank you, everybody, and have a great rest of your day, and we'll see you tomorrow.